Let's start. Hello, everyone. I'm Guili Zhao. I'm the automation application scientist from MGI. Today's topic is automated library preparation, efficient solution for massive parallel sequencing. This is the content of this webinar today. At the beginning, I will give a brief introduction of MPS library preparation. And next, I will go through what's the pain points of the manual library preparation and why the automation is the efficient solution. From this image, we can see that the cost of sequencing is going down per year, and from the $100 million per genome at 2001, and decreased to $1K at 2019. This means the sequencing is become more and more affordable. So the demand of sequencing increased obviously. So currently, the MPS or next generation sequencing is a common DNA sequencing technology, which is a high throughput method used to determine and process multiple DNA sequencings in parallel. Here is a typical NGS workflow. Includes the DNA extraction and repair, adapter ligation, purification, PCR, QC, and sequencing. So we can see that usually the library preparation is the bottleneck of the NGS workflow because this process consists of many steps. So it's not easy to prepare the high quality and reliable libraries with high yield. There are also many pain points of manual NGS library preparation. Firstly, it's a labor-intensive process to prepare high throughput samples and handle many reagents. And the library preparation has numerous steps, so it's a time-consumer process, which usually take one single day or even multiple days for some complicated protocols. It also requires high accuracy and skilled pipetting for small volume. Another pain point of manual library preparation is that variabilities between users occurs even for the experienced scientists. Because the small differences in pipetting can produce much differences in library quality and yield. Another disadvantage of manual library preparation is that it is possible to introduce a cross-contamination. So now all the pain points and the challenges can be minimized by automation. Okay. So according to how the automated machine works to replace the manual experiment? Maybe some audience has not much idea about automated library preparation. So I will introduce in detail here. Usually, the automated machine integrates multi-modules with different functions to conduct the manual operation. For example, the important part is a robotic arm with 8 or 96 channel pipette to transfer and handle the liquid with a high throughput and with a high precision. And for MGI automated system, the pipetting accuracy, the CV is less than 5% for 2 microliter, and is less than 1% for 200 microliter. And usually also the automated machine integrates a magnetic rack to do the compatible with 96 well plate to do the DNA purification and the size selection to using the magnetic beads to replace the manual work. Okay, there is a thermal module for the 96 well plate on the MGI automated machine, which can store the reagent such as the enzyme and the buffer at 4 degrees to replace the ice box. And the MGI automated system also integrates a shaker to do the fast, efficient plate mixing, which can replace the manual, the vortex mixing by manual. Besides the automated machine, automated pipette also can perform the tip mixing, which can work well for the large volume and prevents the cross-contamination. And we know the NGS workflow usually includes many steps of enzyme and PCR reaction. So the MGI automated machine integrates an on-deck thermal cycler called ODTC to replace the external PCR machine. The comment of the open door or closed door or the PCR method can be conducted automatically, and no need to transfer the sample again. So the ODTC is compatible with the common PCR labware and has a sealing lid to prevent the evaporation. And the consumables can put on the deck positions, such as the 96 well plate, to replace the tubes for the high throughput. And the system also has a gripper to transfer the plate to another position. For example, from the magnetic module to a shaker for the DNA elution to replace the manual transfer. So the MGI automated machine can provide the fully automated solution for NGS library preparation. So according to the introduction, we know that there's a lot of advantages of automated library preparation. The automation can improve the throughput and productivity. So you can run up to 16 or 96 reactions per run. Depends on the automated system and the protocol. The automation also can help to reduce the time consumption and enhance the cost efficiency. The automated workstation will transfer the liquid with high accuracy and precision in different pipetting mode and parameters for the different kinds of liquid. Another advantage of automated library preparation is that it can decrease the error rate and achieve the high reproducible results. This situation is usually occurred in the lab that the pipette of user may make mistakes if any interruption and or conversation during the pipetting process. But the automated system can address this issue because they will not forget all the information. On the other hand, the system can record a lot of information to allow you to track the samples, such as what happened from the samples and how much volume of reagent added and the running time and so on. And the automated system also can prevent the cross-contamination because it integrates a HEPA system and a UV lamp to do clean before and after the experiment. And the MGI automated system also can perform the contact-free dispensing with the filter tips. Here is a general automated workflow to show how to run an automated library preparation. Firstly, set up the software and select an application. The only one thing the user have to do is to add the reagent and samples and place the consumables on the deck, and then click to run the workflow. And finally, you will collect high quality and reliable libraries. Here is the easy to use software of MGI Automated Machine, MGI SP100. Okay. MGI Automated System is also an open platform and has a flexible control software like this. So the user can select the application here and place all the consumables on the deck and click to run. And the user also can check the log information of the robotic activity at this right side. Okay, so let's check. Let's see the performance of the automation application by some examples. We know the NGS workflow has several types of DNA purification. So let's see the results of DNA purification by automation. From this figure, we can see that the average recovery rate of is higher than 85%, and the CV is less than 10% for almost the 300 samples. So we can collect the high quality and stability purification results on the MGI automated system. And it will take only 35 minutes for 96 samples purification. The data is based from it's on the MGI SP960 automated workstation. As the NGS development in medical area, the NIPT, the non-invasive prenatal testing, is becoming more and more popular. It's a vital and useful method to determine the risk that the fetus will be born with certain genetic abnormalities from the cell-free DNA. Here is the library preparation process. We can see that there are several steps to prepare the library, so it's not easy. And we can see that it will take one working day for manual operation. And but for the automated library preparation, the process has much shorter turnaround time, and only 15 minutes hands-on time. That's the result of library preparation for NIPT. Taking the 48 cases of fragmented DNA and 212 cases of pregnant women plasma as examples. So we can see that all the libraries can collect the high yield for the sequencing. From the sequencing results, this right table, we can see that the correct detection rate of each sample is 100%. So the MGI automated workstation can provide the reliable and high quality libraries. Okay, and there's another examples for library preparation for microbial or pathogen detection. And also the process includes many steps. And we can see that the sample preparation turnaround time is 5 hours for automation, which is much shorter than 8 hours of the manual operation. And besides, the automated workstation can provide high productivity. Can run the 96 samples per run. Okay, here is a result of the library preparation for the pathogen detection. From the first figure, we can see that all the stable and reliable library can be obtained from the virus samples type on MGI automated system. And after the sequencing, the mapping rate of the automation is very similar with the manual operation. And for the all 24 clinical samples, the final pathogen identification results are exactly the same as manual results. We also have developed some applications using the third-party reagent kit on MGI automated machine. Here is an example of the library preparation for the cancer panel using the pathogen reagent kit. And the turnaround time of automated sample preparation is only 6 hours with 30 minutes hands-on time, and while the manual operation will take 10 hours. The results show that from the first figure, we can see that the library are stable and qualified in concentration for different samples. And the capture rate of the automated operation is higher than the manual operation. From the last figure, we can see that the mutation rate of automation are similar with that of a manual operation. In conclusion, the automated machine can provide the high quality and reliable libraries. Okay, at last, I will introduce the automation products customization from MGI. Currently, we have 2 sample preparation systems launched, but not only restricted in NGS workflow, but also can be applied in general liquid handling. The MGI SP100 has 8-channel pipette and 6 SBS positions on deck, which can prepare up to 16 samples per run. And MGI SP960 integrates a 96-channel pipette, has 24 SBS positions on deck, and it can prepare 96 samples per run. MGI also has 2 NGS workstations for both library preparation and sequencing, which is MGI FP SL200 and L200. They are the one-stop solution from the blood sample or cfDNA to the final sequencing report. So they can be applied in the pathogen fast identification and can prepare the 16 samples per run. And another fully automatic high-throughput blood processing system called Raptor. It can perform the automatic plasma separation and buffy coat separation. and with a high throughput at 1,002 samples per hour, per 8 hours. So all the MGI automated system are the open platforms for customization. Depends on the specific application. Generally, we will collect the requirement and the demands from the customer, and then we make the whole solution for the application and write the script if need. After the feasibility test, and we will run the real sample test and provide the report and the instruction with the training. Here shows some applications was developed on the MGI automated products using the MGI reagent kit. And it also has developed many applications on MGI automated products using the third-party reagent kit and provide the customized solution. And the application collection is becoming more and more. So welcome to give us the application, and we will provide the whole solution for you. Okay, so if any question, if any you would like to get more information about MGI, so please contact us. Okay, so that's all the content.